So, hello everyone. We are here in our video blog of the South American Mycorrhizal Research Network. What we are reading is the name of the blog. And today we have a great guest, Dr. Francis Martin. He works at INRA in France and also at the Forestry Beijing University. It's a great honor to have you. Dr. Martin has worked with mycorrhizae since the 80s, I guess.
Yeah, I'm
Um,
getting old.
and today he will talk us about a little bit about a recent genomic paper in Nature Communications on the topic of mycorrhizal evolution and transitions from saprophytic states to ectomycorrhizal states. So it's a great recent paper. So Dr. Martin, is a great honor to have you. And can you start telling us a little bit about how this study came about, who financed this study, who was involved?
Yeah, of course. First, thank you very much for the invitation. It's a great pleasure for me today to talk to you, but in the meantime to talk to the fungal community, and of course I'm talking today on behalf of a very large team of people. More than 50 people were involved in this comparative genomics, large-scale comparative genomics study. And this research, in fact, started more than 10 years ago now, in 2011, after we published the work on the genomes of Laccaria bicolor and the genome of, the black truffle of Perigord, Tuber melanosporum. The Joint Genome Institute, with which we are collaborating, and we're collaborating on these two genomes, was very happy. And so when submitted the proposal to sequence 25 genomes of mycorrhizal species, including ectomycorrhizal species, but also ericoid, orchid, and arbuscular mycorrhizae, the application was funded. And so we started to sequence 25 genomes at the very beginning. And then step by step, we move up to more than now 150 genomes. So in 2015, we published the first chapter of this long story in Nature Genetics. And so we did in 2015 summarize our work on about 12 genomes. And then we keep working and we keep sequencing genomes more and more. And so the paper we published in Nature Communication a few weeks ago is, I would say, chapter 2 of this long story. And so the work was funded by the Joint Genome Institute, which is a branch of the US Department of Energy. It's about a few millions US dollar funding. And of course, when the genomes are sequenced, you need people to analyze them. So many postdoc researchers were involved in this work. And for this recently published work, most of the work was done by Shingo Miyoshi, one of my postdocs, who was funded by the Lab of Excellence Arbre that I'm heading there in Nancy. And so, yeah, it's a big scale study involving many groups in the US at the JGI, all the people doing the sequencing, assembly of the genomes, annotation of the genomes. And then these genomes are transferred to the different labs, mainly David Ibet at the University of Clark in the US, László Nagy in Hungary, and here in Nancy. So altogether, 3 postdocs were involved in the study for the last 3 years, and many collaborators, including people working on the carbohydrate active enzyme that we are going to talk after. So it's not only my team work, it's really a large scale study involving a lot of people collaborating and having exciting time sharing results.
It sounds like too much work. It sounds like a lot. And you just say that this was chapter two. So are you heading to a chapter three, I guess?
Yeah, we hope so. Now that we are aiming to sequence 300 genomes of mycorrhizal species. Again, not only ectomycorrhizae, but we are focusing also on ericoid mycorrhizae, orchid mycorrhizae. We would like to have more arbuscular mycorrhizal genomes because, in fact, if we already sequenced many genomes, it's only a small proportion
Yes.
of the mycorrhizal genome. So all the conclusions we've got from this sequencing should be confirmed by having a larger set of species with a more ecological diversity. So
Uh,
yes, maybe there will be a chapter 3
Fantastic.
soon.
Fantastic. In the introduction, you mentioned that mycorrhizal evolution could be convergent. Can you give us some examples of this convergent evolution?
Yeah, of course. For people working on let's say ectomycorrhizae, if we focus on ectomycorrhizae, to me, when I was a young scientist, and I'm sure it's the same for you now, I've always been amazed by the fact that all ectomycorrhizal roots will have the same anatomy, the same structures. You will find the mantle sheath around and sheathing the root tips. There's the artignates with the hyphae penetrating within the roots and interacting with the root cells. So if you induce mycorrhizae formation by using Lacaria bicolor, the basidiomycetes, or Tribermelanosporum or Sinococcum geophyllum, which are ascomycetes, you will get the same structure. The root tips, the mycorrhizae, the ectomycorrhizae, will present the same structure with the mantle and the artignates. So that's what we call the morphological convergent evolution. And if you use 10 species of mycorrhizal fungi, ectomycorrhizal fungi, 1,000, you will get mainly the same structures: extramatrical hyphae, the mantle sheath, and the artignate, independently of the taxonomic level, independently on the clades. So either ascomycetes, basidiomycetes, agaricales, boletales will establish the same kind of structure. And if you go deeper in the mechanisms, the physiological mechanisms, the biochemical mechanisms involved in the mycorrhizae, all these mycorrhizae, ectomycorrhizae, will involve the transfer of carbohydrate from the plant to the fungus and phosphorus and nitrogen from the fungal part to the root, to the plant parts. So again, it's an amazing convergent evolution at the physiological level. So we were wondering whether this convergent evolution was the result of some common genetic programs.
You also talk quite a bit about the transition between saprophytic states to ectomycorrhizal states. Before doing the analysis, which were your expectations? Which were your hypotheses regarding this transition?
We knew from the work of previous scientists, like Leo Tedozo, many years ago, published a very, very fascinating paper showing that using ribosomal DNA and different phylogenetic markers, they showed that the ectomycorrhizal symbiosis evolved from saprophytic species many times during the evolution of fungi, probably 60 to maybe 80 times. And all along the tree, including ascomycetes, basidiomycetes, the different types of basidiomycetes. So we knew from this work based on phylogenetic markers that the transition to ectomycorrhizal symbiosis evolved from saprophytic ancestors. But no one knew what was the mechanism or what were the mechanisms involved. So our task was really to look at the genetic programs involved in the transition, how saprophytic fungi, wood decayers, soil saprotrophs, how they can one day switch to establishment of the mycorrhizal symbiosis. What are the mechanisms involved? So this was really the driver of this research. Sequencing genomes on a large scale, sequencing genomes of phylogenetically relevant species, making a good choice along the tree of life of ascomycetes and basidiomycetes in the main clades of fungi, knowing that many of these clades play a key ecological role. And then we started to sequence these genomes. And not only sequencing the genomes, you need also to get information on the expression of the genes. So all these genomes have been sequenced, but when we could, we also produce transcriptomic data. So not only having the repertoire of genes, but also looking at the gene expression during the interaction and the combination, the integration of large-scale genomics and large-scale transcriptomics allowed us to decipher the mechanisms involved in the transition. But in fact, the result, your question was, did we expect the results? I should say no. I mean, when we started the work more than 10 years ago, I expected to find genes, fungal genes involved in the communication with the plants to allow the establishment of the symbiosis. But we didn't expect that the transition from saprotrophy to symbiosis would involve the loss of genes, specifically the loss of genes involved in the wood decay or polysaccharide decay or the plant cell wall decay. This was really one of the main unexpected results.
Could you say that that was the main mechanism, the main genetic mechanism, and the main finding of your study? This loss of genes involved.
If I should summarize, I would say that the main mechanisms allowing the transition from saprotrophy to ectomycorrhizal formation likely involved at least two molecular mechanisms: the loss of enzymes involved in the polysaccharide degradation. So loss means that all the genes or most of the genes coding for cellulases, hemicellulases, ligninase, so lignin peroxidase, are lost in the clades of ectomycorrhizal fungi. Sometimes all of them are lost. So this means dozens of genes involved in polysaccharide degradation, wood degradation, organic matter degradation, they are lost during the transition. And then second mechanism, to enter the roots, this fungus should be able to control the plant defense mechanisms. So what we found is that many genes induced during the mycorrhizal formation are in fact small secreted proteins that are injected by the fungal mycelium into the root cells. And we showed that some of them are moving to the plant nuclei and control the defense reactions. So you see, these fungi, to interact with the plants, they need to lose the plant cell wall degrading enzymes, probably to avoid destruction of the plant cell wall, of the host cell walls, and likely this reduce the defense reactions. So that's probably a prerequisite to enter the plants, but also they need to have specific genes to control the plant defense reaction. So I would say that the two main mechanisms are there. But
Are these two mechanisms always like coordinated and they are equally like distributed across, for example, Basidiomycota and Ascomycota, or are there like phylogenetic differences between these mechanisms?
we found these mechanisms, these two major mechanisms, loss of plant cell wall degrading enzymes and acquisition of new genes to control the defense reaction. We found that two mechanisms in all species we have investigated so far. So it's really one of probably the major drivers for mycorrhizal formation. But what we showed in this new study is that when we focus on the early diverging clades of fungi, uh, like the Cantarellales or the Russulales, so very ancient clades of ectomycorrhizal fungi, we've been able to find a few of them uh, which still have some enzymes. So, and this, some plant cell wall degrading enzymes. So they're still at, I would say, at a very early step of the symbiosis evolution. They still have some of these enzymes, cellulases, pectinases. But during the interaction with the plants, these few species having kept these enzymes, they repress the enzyme at the transcript level. So the genes are there.
Mhm.
They have not yet lose these genes during the evolution, but when these fungal hyphae interact with the plants, these genes are turned off during the interaction. So it's a, it's, of course, it's speculation, but I suspect that the first step of the ectomycorrhizal interaction has been possible because some of the soil saprotroph still having these plant cell wall degrading enzymes turn these genes off to be able to enter the root cells and then communicate with the plants.
That's incredible because um, there are currently like big debates about what exactly or how do you define an ectomycorrhizae and even an arbuscular mycorrhizae, if is by functional traits or is if is by just the morphology, like the heart thickness and a lot of that. So I think that this research can boost this debate, can give like some light into that debate because that thing that you say about the genes switching off when they interact with the host,
Mhm.
that's truly amazing. Um,
Yeah,
coming
so
back, yeah, sorry,
just to finish up that part of the discussion,
yeah.
I think now the next step. So we've been able by sequencing more than 50 genomes of ectomycorrhizal fungi. We have been lucky enough to find a few ones where we can see the transition in action. They still have some saprophytic ability, but they are already symbionts. So now we are aiming to sequence more genomes from these early clades to try really to decipher step by step this transition from soil saprotrophy to symbiosis. So
Wow.
I think evolutionary speaking, it's probably one of the key events in the life of fungi. And so we really need to find people able, I mean, that's one point I would like to emphasize. Now sequencing is very easy. I mean, sequencing a genome at the JGI, it takes a few weeks to get the annotation. So the real bottleneck today is to get samples, to get cultures, to get fruiting bodies, mushrooms of rare species. And then it's getting more and more difficult, in fact, to extract DNA and to get a high quality DNA. So the bottleneck now is not sequencing, gene prediction, and analysis of this large scale set of genomes. It's really, to me, the most difficult part now is to find to decide to work on this clade, to find someone having the culture or mushroom to extract DNA. So my call to the community is really, if you have interesting mushrooms, interesting species of mycorrhizal fungi, just let me know. And then we can try to work together to extract DNA from these early diverging clades.
So what do you mean by interesting? Like representative of the whole phylogeny or ecologically representative?
Ecologically relevant. For example, we need more genomes from the Cantarellales.
Mhm.
There are only a few. We need more genomes of Telephorales, Sebaceanales. So we have only one or two genomes. It's not enough. So I think that if we can promote the collection of these early clade species, this would be really great.
Fantastic. Just coming back to the figure two on your article, uh,
Mhm.
it was quite striking to me because here you show the size of the genomes and especially the arbuscular mycorrhizae, but also the ectomycorrhizae, the genomes are quite big in comparison to other like fungal guilds. Uh, can you elaborate on this? Why these genomes are so large? What that means? What are like the
So when
explanations of this?
we found this striking difference in the size of genomes, of course, we checked for whole genome duplications, which is a very common mechanism in plants to increase the size of the genome. In fact, in this fungi, we never been able to find any genome duplications. So the main explanation for the increased larger size of the genomes of most, at least the ectomycorrhizal and the arbuscular mycorrhizae, is the content in transposable elements. Most of these genomes are very rich in transposons, uh, as you see on these figures.
Mhm.
And so some of the largest genomes sequenced so far in fungi, like Gygaspora, the arbuscular mycorrhizal species, is really 800 megabase, and 90% of the genome is made of transposable elements. In ectomycorrhizae, we have species like Cinococcum geophyllum, 300 megabase, or Tricoloma matsutake, more than 150. Again, most of the genomes of these genomes is made of transposable elements. Many families, as you see on this figure, but mainly long terminal repeat LTR transposable element like gypsy and copia. They are really among the most abundant family. And so the question is why? Why have we so large quantity and families of transposable elements in mycorrhizal fungi compared to saprotrophic species? In fact, we have no explanation yet. That's clear cut. We have most of the genome sequence so far are much larger. They are much larger because they are full of transposable elements. But why? It's some people could say, of course, then it's mainly speculations. First speculation, we could assume that ectomycorrhizal fungi or mycorrhizal fungi, they live in the soil, they live in planta, they interact with many plants, so their genome should be very plastic.
Mm.
And a good way, we know from previous studies in animals, in insects, in planta, that transposable elements are a driver for gene innovation.
Mm.
Because when the transposable element, this repeated sequence jumps along the genome, they can produce mutation, reorganization, they can create new genes. So many people think that transposable elements, are one of the main driver of gene innovation.
Mm.
So of course, for a fungus like Aria or like any of the ectomycorrhizal fungi, they live in a very variable world, a very plastic environment. So maybe evolution have favored these transposable elements to generate new genes which allow these fungi to interact with plants and sometimes with many plants. If you were talking about arbuscular mycorrhizae, they have the largest genomes we have sequenced so far. And we know that Rhizophagus or Gygaspora, they do interact with hundreds of different species of plants. So maybe these transposons are used or are one of the main reasons why there are many genes in arbuscular mycorrhizae, 28,000 genes, and this large set of genes may allow these fungi to interact with many plants. But
Do you
again,
know,
it's speculation.
do you know if these transposable elements are so common in, for example, in lichens or in coral reefs, in other symbionts?
I don't think so. Very often, at least for example, in the symbiosis between insects and bacteria, it's in fact a reduction in the genome size. Many of the symbionts, the bacterial symbionts of insects, like Buchnera, their genome is shrinking because they live in interaction with the host, and then they lose some of the function. So we were really amazed by these results when we got the genome of Lacaria bicolor and the genome of Tuberman anasporum more than 10 years ago. These genomes were big, are big, and we were very surprised to see this large difference. So there's more work to do. So there's plenty of work for a new generation of scientists. Anyone interested by this can use this resource for that.
I have now a double question. You mentioned that you use a phylostratigraphic approach, and by using this approach, you found that I will read in Ascomycota and Basidiomycota an average of 74 and 67% of the ectomycorrhiza induced genes predated the evolution of the ectomycorrhizal symbiosis, respectively.
Yeah, yeah.
This is also something very amazing. So can you tell us about a little bit about the phylostratigraphic approach and this result?
Yeah, in fact, there we use two approaches based on the transcriptome data to decipher the origin of the symbiosis regulated genes. So the basic principle of this approach is to, in fact, using transcriptomics to identify the genes induced or repressed by the symbiosis. And then we look at the conservation of the protein sequences throughout the tree of life, getting back to the origin of basidiomycetes, ascomycetes, and fungi. And so this double approach, this dual approach, let us know how these genes evolved. And then we found that mycorrhizal interaction, or at least ectomycorrhizal interactions, require, in fact, involve two sets of genes. Genes in what we call symbiosis-induced or symbiosis-repressed genes. So let's call them symbiosis-regulated genes. Two-thirds of them are conserved. So this means that when we compared the protein sequence to all the fungi, including saprotrophic species, wood decayers, soil saprotroph, these genes already exist in these species. So they are very ancient genes. And their sequence is well conserved. And very often they code for primary metabolism pathway, so glycolysis, or they code for membrane transporters. For example, many of the transporters involved in the carbohydrate transfer between the two partners, they already exist in the saprophytic ancestors. So this means that these genes, pre-existing genes, some of them very old, already existing before the split between Ascomycota and Basidiomycota,
Wow.
these genes, old ones, conserved, are in fact getting a new function during the interaction. And this new function is really important for the symbiosis. So I call that neo-functionalization. So old genes, but getting a new function, which is playing a role in the symbiosis. So about two-thirds of the genes regulated by the symbiosis already existed before the symbiosis emergence. So again, most of them are genes with a well-known function, transporters, enzyme involved in the nitrogen assimilation or carbohydrate assimilation, some structural protein like hydrophobins. So to make mycorrhizae, the fungi used material, pre-existing material, to construct the symbiosis. But still, one third, between 5 and 30% of the genes are specific to mycorrhizae, specific to symbiosis, and only existing in the ectomycorrhizal fungi. And we showed that each clade, bolides or Lacaria or the black truffle, each clade will encode about 10 to 15% species-specific genes, but these species-specific genes involved in symbiosis are very different from one clade to another one. So each clade of fungi able to establish mycorrhizae have evolved its own set of species-specific genes.
Wow. Yes, you mentioned that you, from like almost 6,000 symbiosis-induced genes, about 50% of them, 15% of them are shared between all species,
Yeah.
and like 31% are these species-specific genes, which is also very interesting result.
Yeah,
And
some genes are really conserved. 15% of the genes are conserved throughout the tree of life. So from Glomeromycota to Ascomycota and Basidiomycota, we found these 15% genes of the symbiosis-induced genes in the three main phylum. Some genes are conserved, but only in Basidiomycota and Ascomycota, and some genes are only conserved either in the Basidiomycota or the Ascomycota. So we have all, it's like a step-by-step evolution.
fantastic. Um, can you tell us, to finish, can you tell us a bit about, because the article mainly talks about this transition and mainly talks about ectomycorrhizae, but can you tell us about the results in ericoids and orchioids and arbuscular mycorrhizae?
Yeah, of course. In fact, we have published last year, or maybe 2 years ago now, a paper comparing ericoid genomes. And they are strikingly different. I mean, that is really a different story. The genomes of the ericoid species is very different from the ectomycorrhizae. In fact, most of the ericoid fungi we have sequenced so far are very similar to the genomes of saprotrophic species, of soil saprotrophs. They have a large set of plant cell wall degrading enzymes. Even in most cases, Rhododendron, Melaniomyces, which are well-known ericoid fungi, model systems for ericoid fungi, these guys, they have the largest set of plant cell wall degrading enzymes, even larger than many of the soil saprotroph.
Wow, wow.
So I think these guys are really saprotrophic species still. So when they live in sphagnum, in mosses, they are able to degrade these mosses and sphagnum substrates or litter substrates. But at one step, when they do interact with the plants and when they start penetrating in the root system, some of these enzymes are used to penetrate within the root cells to form the mycorrhizal structure, the pollutants. So it makes sense for them to keep some of this enzyme to really grow in the apoplastic space and penetrate within the cells. But many of these genes are again turned off at the transcriptome level, at the transcriptional level during the interaction. So again, even if they have kept a large set of these plant cell wall degrading enzymes, many of them are switched off during the interaction.
Oh.
So it's a striking difference between, I see there a very dramatic difference between ericoid and ectomycorrhizae. They really behave very differently. And I would speculate that it's probably due to the fact that ericoid symbiosis is considered as younger than the ectomycorrhizal symbiosis, and even younger than the arbuscular mycorrhizae. So if, roughly speaking, arbuscular mycorrhizae evolved 450 million years ago. This is the oldest symbiosis. Some people say the mother of the symbiosis. Ectomycorrhizal fungi evolved with the Pinaceae 180 million years ago. And ericoid fungi, they evolved with the Ericaceae only 100 million years ago. So I suspect that along these gradients, the arbuscular mycorrhizae, they have lost all of the plant cell wall degrading enzyme. They have the lowest repertoire. Then we have ectomycorrhizae, where we discussed already that there is a wide variation, diversity. Some of them have lost most of the plant cell wall degrading enzyme. Some have kept. And the ericoid, they have a very large set of enzymes. And this is the same for orchid mycorrhizal fungi. Again, like the Sebacina species, they have a substantial repertoire of plant cell wall degrading enzymes. So when we say mycorrhizae, we should really think differently, at least at the genome level and genetic mechanisms between ericoid, orchid, arbuscular, ectomycorrhizae. They have evolved different mechanisms, and the evolutionary steps are very different.
And almost to finish, you at the end of the article mentioned that it's a big question, but ectomycorrhizal lineages have emerged independently many times. You mentioned like 80 or something like that.
Yeah, 80 times at least. Oui.
Why is this so common? Why this happens? It's a big question.
That's a difficult question, of course. Let's say, I mean, being a mycorrhizal fungus is providing a lot of benefits to a species, because in fact they are living within the roots, so they are protected from the outside competitions. As you know, the rhizosphere, the surroundings of the roots is a highly competitive world with a lot of species fighting, releasing nasty compounds. So being a host, living in host, being protected from the outside is already a key evolutionary step. They have a nice home where they can survive, and then they are fed by the plants with carbohydrate, pure sugar, sucrose or glucose. Instead, in the soil, they have to use these enzymes, these degrading enzymes, to decay the organic matter. So I would think that living in interaction with a plant is providing a lot of evolutionary benefits. And the holobiont made of the plants and the fungus, and even a cortege of several species of fungi is a very efficient way to evolve and to survive in a very competitive world. Cooperation is likely, to me, is a key step in the evolution of plants and fungi. So being to conclude, it's fun to be a mycorrhizal fungus living inside a nice plants and being fed by the plants.
Yes, I totally agree that cooperation is key in this.
Yeah.
Something else that you want to add?
I think I already talked a lot, so I think again that I would be very pleased, very happy to collaborate with people collecting fungal material in the field. We need more samples, we need more culture, we need more DNA to sequence and to generate these genomic resources, not only for describing the genomes or deciphering the mechanisms. I think these genomes can be used now for molecular ecology. We can set up meta-transcriptomics projects where we can go everywhere in the world, collect the RNA, and then quantitate the expression of genes in situ, in the forest, in the fields, by using these genomes. But again, there are 20,000 species of ectomycorrhizal fungi, and we have sequenced only 200. So my dream would be to sequence 10,000 species of ectomycorrhizal fungi, ericoid fungi, orchid fungi, arbuscular mycorrhizae, because these genomic resources can be used to better understand the ecology of these fungi. So I like this idea of coupling more tightly, coupling genomics and molecular biology to ecology. That's the way to go, I think, for the new generation of scientists.
Fantastic. Thank you very much, Dr. Martin.
Thank you to you.
It was a great pleasure and see you everyone soon.
Bye.
Bye.